Hello, class. Alright, so after learning about fish as a trade commodity and the fish as a food commodity, we are now going to study fish as a perishable commodity. So we will, this module actually, no, module 3 is divided into two parts. So the first part will be on the pre and post-mortem changes in fish. And the second part will be, we will be discussing about the agents of fish spoilage. Okay. So to start, I hope you can see my screen. No? I'll just turn off my video because up to this time I am not feeling well yet, and I don't want you to see what I am doing in front of my computer. Okay, I'm doing a lot of extracurricular activities. Okay, right. So this particular slide shows the wait. Why is it that it jumped in here? All right. So we know that fish, while they are living, no, their muscle remains sterile. All right. So as soon as they die, they undergo enzymatic, biochemical, and microbial changes, and that eventually lead to the invasion of microorganisms up to its muscle. So the statement that fish muscle is sterile is only true when the fish is still alive. All right. Although there are indigenous microorganisms that are present in the skin, in the gills, and in the gut of fish, no, its muscle remains sterile as long as it is living. All right. So as we go on with our discussion, you will learn that microorganisms will invade the tissue of fish during autolysis, no? And eventually it will cause a lot of changes in the biochemical components of the fish. Okay. So this slide summarizes the events, no, that's happening in the fish pre-rigor, during rigor, and post-rigor. Okay, so as mentioned, no, as mentioned, fish remain sterile while they are living, and upon death, no, there will be no more supply of oxygen that will support the need of the tissues in the production of ATP. So the fish will start to go into rigor. Okay, so the onset of rigor mortis is dependent on the initial amount of ATP in the fish tissues, no, before its death. So later you will know that the onset of rigor mortis is affected by several factors. So when you say rigor mortis, now the fish is undergoing stiffening of the muscle. So since there will no more oxygen that will supply the need for aerobic respiration, the pathway now will shift into anaerobic glycolysis, wherein glucose will now be used to produce ATP, but the ATP level that will be produced is at much lower concentration as compared with aerobic respiration. All right, so there will be reduction in the ATP level, and since the pathway now that will be taken is anaerobic glycolysis, there will be fewer amounts of ATP molecules that's being produced, and there will be accumulation of acidic byproducts, and that would include your lactic acid. So it is also known that once the fish dies, its muscle tissue will eventually turn into slightly acidic. All right, because of the accumulation of lactic acid. Okay, after rigor mortis, it will now proceed to a stage known as resolution of rigor. When you say resolution of rigor, diba, at pre-rigor stage, the fish is soft, no, and elastic. At rigor, it becomes stiff. During the resolution of rigor, it turns back into a soft state ulit. So it will return back to its being soft, no? So after that, it is now prone to a lot of changes, no, degradative changes, and that will include autolytic spoilage acted on by the proteolytic enzymes, no, and then there will be ATP degradation, and then there will also be microbial changes, no, that will be caused by the proliferation of microorganisms. So because of the autolytic spoilage, no, there will be the gut and the other tissues will be ruptured by the action of the proteolytic enzymes. Now the microorganisms can freely invade, no, the previously sterile muscles of fish. All right. So there will also be degradation of TMAO. Okay. And at a later stage, there will be lipid peroxidation and the production of free fatty acids. And of course, all of this will be manifested in the sensory quality of the fish, wherein there will be off odor, off flavor, and eventually degradation of the obvious degradation of the fish tissues. Okay. So to summarize, no, the post-mortem changes in fish should include sensory changes, changes in its smell, taste, texture, appearance, and color. There will also be enzymatic changes, no, specifically autolysis or self-digestion acted on by the proteolytic enzymes. Next would be under enzymatic changes would be your ATP degradation. So a lot of catabolites, no, will be produced that would be beneficial for microbial growth. Okay. Another would be changes caused by microorganisms. So there will be a lot, you know, of factive changes that these microorganisms can cause to our fish. And also there will be chemical changes caused by lipid peroxidation and hydrolysis. No? Okay, so first for sensory changes. Oh, wait, my slide is not moving. Okay, so these are the these are the sensory changes in fish, no? So you have to observe the eyes, gills, odor, flesh, the slime, presence or absence of slime, the belly walls and scales, no? So for fresh fish, these are the characteristics of a fresh fish, no? So eyes would usually be bright, full and bulging, while in stale fish, spoiled fish, it looks dull and sunken. Another obvious manifestation, you know, your mother would usually check if they buy fish in the market. They check the gills if it's bright red, no, and covered with clear slime, and it doesn't have an off odor. So for the stale fish, the gills would look dull, brown to gray, slimy with offensive odor. Other than odor and flesh, of course, for the flesh, you have to press it gently. Of course, don't press it as if you are pressing something, no, na you just have to press it, because the vendor would of course react if you sort of medyo inabuse nyo yung muscle ng fish nila. Okay, so if that leaves finger impressions on the muscle, that would indicate na the fish is already stale or spoiled. Aside from that, the eyes, gills, and flesh, another obvious thing to check whether the fish is still fresh or not is the belly walls. If it's still intact, it's fresh. If it's already ruptured and showing some openings, no, that would indicate that the fish is already stale. Of course, for scaly fishes, if the scales are hard to remove, that would indicate that the fish is still fresh. And if it's already loose, no, it's already spoiled. So it sounds easy to identify a fresh fish, no, but in reality, it's also challenging. Actually, if we can meet face to face, you will be doing a practical exam wherein you're going to check if the fish is fresh or not. Okay. So next is we have this figure from Haas to 1976. This is a very old study, but we still use this because this result still holds true, no, up to this time. So he used ice cod, no, in studying the eating quality of such fish, and he was able to come up with these 4 phases of changes that's undergoing in the fish, no, during storage in ice. So he indicated here 10 as the absolute freshness, 8 as good quality, no, 8 to 6 good quality, neutral, tasteless if its score is 6, and it's for rejection if the score is 4. Now, the x-axis would indicate the quality score. Parang he used a sensory scorecard with it's a 10-point scale, no? A sensory scorecard. And this one, the y-axis, wait. The x-axis refers to the number of days, and the y-axis refers to the quality score. All right. So what are these phase 1, phase 2, 3, and 4 indicates? So phase 1, no, tells us that the fish is still very fresh and has a sweet, seaweedy, delicate taste, no? And the fish would stay at this condition in the case of cod, no, for 2 to 3 days after catching. Phase 2 tells us that there is a loss of the characteristic odor and taste, and the flesh becomes neutral, but still no off odor that is detectable. Okay, and the texture of the fish is still pleasant. Okay. Phase 3 indicates that there is already a sign of spoilage and a range of volatile and pleasant smelling substances is produced depending on the species, no? And that would be an indication that volatile compounds such as TMA, no, derived from the reduction of TMAO is already accumulated in the fish. Okay, especially if the fish has initially high TMAO. Okay. Phase 4, of course. Phase 4, as you would see here. Wait, I can't move my... Yeah. Phase 4 here shows that the fish is already putrid and stale. So as you can see, nag-drop na yung graph niya, no? From day 0, 1, 2, before the day 2, its score is above 8, meaning it's still fresh. At day 2 to 6, it drops to around 6, score of 6. So it already have undergone autolysis, no? And at phase 3, day 6 to 12, it's nearing rejection. So at phase 4, day 12, ni-reject na yung iced cod. So with this graph, this shows that the eating quality of iced cod is still good up to day 6 at ice. Actually, it's best at day 2, and it's still acceptable up to day 6. But as it increases the storage time up to day 12, it's already for rejection. Okay? So what's the importance of knowing this? No? So you would know how long should you keep your fish in ice, no? In chilled condition. So as much as possible, you keep it for 2 days or less. Okay? And you already have to process or to cook it. So you can still avail or benefit its good eating quality. Okay? Okay. So what is rigor mortis? No, this is a very popular term, no, that we've learned back in high school. So rigor refers to stiffness and mortis of death. Okay, so this is characterized by a succession of muscle contraction exhibited by muscle stiffening after death. There is a question in the quiz where I asked, no, what's the biochemical explanation of rigor mortis? And the one assigned to answer that question gave us a very comprehensive answer. So please take a look at that item there, no? So you would also know that certain molecules, no, has or play a role in the contraction of muscle, right? So rigor mortis is attributed to the dynamics of ATP and ADP. The first one is ATP and ADP within the muscles, accompanied by lactic acid accumulation and the fall of pH. So the moment that your ATP drops to a certain level and your lactic acid reach certain level to cause a decrease in its pH, rigor mortis will onset or will start. Okay. This would result to several changes in muscle quality. Okay, so take note that the stiffening of the muscle is gradual and is not, it does not happen in the whole fish at the same time. So usually it starts at the tail and then it gradually goes up to the whole body of the fish. So the first part that's starts to stiffen is the tail. Okay, so there is a pre-rigor. It's still limp and elastic. It becomes stiff and hard in rigor, and post-rigor it becomes limp and elastic again. So that's the resolution of rigor mortis. Okay, so this is how you, if you want to conduct an experiment to determine how long after death does the fish go into rigor. So the best example to use is your catfish, because diba, we can catch a catfish alive and then you can kill it or wait until it dies. And then you can monitor, right? Kung what time siya namatay. And then you wait how many hours siya after death niya will go into rigor. You will know if it's stiffening because it will look like this. No, it will straighten its body. Okay, it's in rigor. And then you can time also how long will it stay at this condition, and how many hours or ilang minutes until it becomes soft and limp again. Ayan. So, pwede mong matayman na, oh, after death niya, 5 hours after that, it goes into rigor mortis, and it remains in that condition for 2 hours, and then after 2 hours, it becomes soft again. So it's a cute experiment. No, before our students used to carry an ice box with their fish inside na naka position na siya na like this, and then they have to carry that styro box to AS, to wherever room no na may klase sila, until mag-stiffen yung fish nila and they return back to the lab pag natapos na nila yung experiment. So sometimes it takes more than 24 hours for them to complete the experiment. So dinadala pa nila sa boarding house yung kanilang fish sample. So wala, cute yung experiment. Ayan. Okay. So what are the factors no that influences the onset of rigor mortis? So let me check if we have it here. No, okay. So rigor mortis does not affect the whole body, as I've mentioned earlier. So it starts at the tail, no? And the factors that affects rigor mortis are the following. So the species, the size, the temperature and handling, the physical condition of the fish, and the catching and the killing methods. So stunning and killing hypothermia, the fish is killed in iced water, give the fastest onset of rigor, while a blow on the head gives a delay up to 18 hours. So ano yung benefit if you delay the onset of rigor mortis? So, di ba, before rigor mortis, the fish is at its best quality. So, if you can lengthen the pre-rigor state of the fish, meaning you can have an extended period of time to still utilize the fish at its best quality. So, kung matagal mo siyang mabenta, at least fresh pa siya nung nabenta mo kasi na-extend mo yung pre-rigor state niya. Okay? So, in terms of, meron tayo ditong handling, no? The degree of exhaustion of fish, no? Fish that struggled in the net before hauled and gutted will have much less reserve energy. Thus, they go into rigor more quickly because we mentioned earlier, no, that the amount of ATP is or the level of ATP in the tissue of the fish would greatly affect the onset of rigor mortis. Once the ATP level drop to a level that can no longer sustain the energy need of the cell or the tissue, it will now go into rigor. Okay, so fish that have spent its energy prior to catch would have less reserves, no? Less energy reserves. So it will go into rigor more quickly. Small fishes usually go into rigor faster than large fish of the same species. And also, manipulation of pre-rigor fish does not appear to affect the time of rigor, but manipulation or flexing of the fish while in rigor can shorten the time they remain stiff. So pwede the fish that's in rigor state kasi matigas, no? Gusto mo na siyang i-fillet, you can manipulate it, no? I-flex mo siya so that it will soften faster. Pwede yon. But sometimes manipulating it or flexing it can cause finger impressions in the tissue that would actually make it make it inferior in terms of muscle quality. Right? So the fish that has been caught from warmer water go into rigor faster, go into rigor and pass the rigor state faster. Also, species of high lipid content goes into rigor earlier than those or than lean fish species. Okay, and if you're going to compare dark muscle and white muscle, dark muscle goes quite earlier into rigor than the white muscle. Okay. So, rigor mortis starts immediately or shortly after death if the fish is also starved, no? And the glycogen reserves are depleted or if the fish is stressed. So, if kakaspawn ng fish, di ba, we know that after spawning, na-release nung fish yung kanyang energy reserves, no, during spawning. So, newly spawned fishes go into rigor much faster than yung mga hindi pa nakapag-spawn. Okay? All right. So medyo mahaba tayo dito, no? So this slide shows the duration, onset and duration of rigor mortis in various fish species. So what is the lesson that we can get from here, no? From studying rigor mortis, pre-rigor and post-rigor state of fish. So it is important that you keep the fish at the cold condition, no? Right after catch, so that it will go into rigor at chilled condition or at low temperature. Kasi mas nagiging prone na siya into deteriorative changes once it passed the rigor mortis state. Okay, so look at this. The different fish species undergo or go into rigor, and the duration of its rigor is different, no? Per species, and also depending on their condition. Look at this. Stressed siya, no? It was placed in 0 degrees chilled condition. So the time from death to onset of rigor is around 2 to 8 hours. Pero pag mataas yung temperature, mas early siya mag-go into rigor, and the end of rigor is quite short also. However, if hindi siya stressed and chilled, no, the time from death to onset of rigor is longer, and the time from death to end of the rigor mortis is the longest as compared to the rest. So the same din dito. Actually, this holds true sa mga unstressed na species. Mas matagal talaga siyang unstressed and kept at low temperature. It goes into rigor longer as compared to the same species that's stressed and at higher temperature. So, ayan. So, what is the significance of rigor mortis? I've mentioned earlier, no, that if you fillet fish in rigor, no, you are going to produce or you will get a low yield, poor yield, filleting yield after you have done after you have filleted your fish, because the fish is very stiff. Mahirap siyang tanggalin doon sa backbone. And it would also result to gaping. Okay. And also, if you fillet your fish before it goes into rigor, the muscle can contract freely, no, and the fillets will shorten following the onset of the rigor kasi mag-undergo pa siya ng rigor mortis. So this is the amount of shrinkage of the dark and white muscle. All right, that would be the effect if you cook the product pre-rigor, in rigor, and post-rigor. At pre-rigor state, the texture would be very soft and pasty. At rigor state, if you cook a fish that's undergoing stiffening of the muscle, the texture is tough, no, but it's not dry. However, at post-rigor, the flesh is firm, succulent, and elastic. Actually, if you process or cook the fish at post-rigor, basta kakatapos pa lang ng resolution of rigor, there's no problem. The problem would be if medyo matagal na siyang natapos sa rigor mortis niya, saka mo siya pinrases. Okay, that's the time that you'll be having some problems. Okay. Right. So this shows represents the shrinkage of fillet, no? So there will be what? The fillet is cut off before rigor, so the length of the fillet is reduced to around 24%. But if you fillet it after rigor, konti na lang yung i-reduce ng kanyang length. So if you are in a filleting industry, it's either you fillet the fish in pre-rigor or post-rigor state. Ayun. Actually, when we do bangus deboning, parang sinasadya natin na nasa post-rigor na state yung fish natin nagagamitin for bangus for deboning. Why? So that it would be easier for us to remove the bones. Kasi nag-undergo na siya ng post-rigor state, nasa post-rigor na siya. If we use fish that is still in pre-rigor state, very hard to remove yung bones, and the tendency is may iwan dun sa muscle ang mga bones natin. So ayan, that's one of the tip that you can use, no, when you do bangus deboning. So when to freeze your whole fish? Ayan. So these are the advantages and the disadvantages of freezing your fish, no, at pre-rigor, in rigor, and post-rigor. So ayan, you just have to read this, no? And then the next slide will show about fillets naman. So, ito yung mga disadvantages at pre-rigor, in rigor, and post-rigor. So, you'll get excellent quality, possible, no shrinkage kapag na-freeze mo yung fish mo in pre-rigor. Ayan. So, ito yung mga sinabi ko kanina na the factors that influences the progress of rigor mortis. So, high lipid content goes into rigor earlier. Very active fishes show a shorter period of rigor mortis, and the exhausted fish have little or no energy reserve, thus they go into rigor faster. And fishes that have undergone starvation, no, meaning they have low energy reserves as well, they go into rigor faster. So ayan, ang mga nag-struggle, mas madaling mag-stiffen, small fishes go into rigor faster, and poor handling would also affect the time of rigor mortis. And also, fishes collected from warmer waters go into rigor and pass rigor faster than those collected from colder waters. Ayan. So, rigor can actually affect the quality of frozen fish in three main ways. So, ayan, gaping in fillets taken from frozen whole fish, no, it would result to gaping kapag poorly handled yung fish natin. And then there will be drip loss, no, of the frozen whole fish or fillets. And of course, as mentioned earlier, there will be shrinkage of frozen fillets. Ayan. The three undesirable effects that was mentioned can be reduced or prevented by the following. Now, keeping the fish cool, particularly before it goes into rigor, no? Now, only careful treatment of the fish before and during rigor will result in a higher quality frozen product. So, for appropriate handling, careful labeling scheme for frozen fish is necessary that enables processors to identify saan yung frozen pre-rigor, frozen in rigor, and frozen post-rigor. That's important and reliable, and the safest way to avoid negative impacts of rigor is to keep fish chilled at every stage before freezing. So kung hindi ka pa ready to freeze your fish, you have to at least keep it cool, no? So you use ice or put it in chilled condition. If the fish pass through rigor at chilling temperature, the effects of rigor on quality will not be so serious. Okay? Okay. So the next is autolytic changes. Okay, so what do you know about autolysis? Okay, so autolysis refers to self-digestion. Okay, so fish contains what you call autolytic enzymes, and most of them are your proteases. Okay, so we have the trypsin, chymotrypsin, and we also have catepsin and your calpains. And this results to the production of spoilage substances that can eventually be detected by sensory evaluation. Okay. And also, it produces metabolites that are capable or that are suitable for growth of microorganism. I can find my cursor. So that's the result of autolysis. Okay, so it starts with careless handling of fish or when the fish cell is broken. That's the time that these enzymes get out of the cell and attacks nearby cells and tissues. Okay, and it results to autolysis. Okay. So this slide also summarizes the events that's taking place in the fish muscle as a result of autolysis. So we know, parang paulit-ulit tayo nito dito, that glucose is used during anaerobic respiration, na it produces a few or very small amount of ATP molecules, and then it accumulates lactic acid that results to the reduction of pH, and then it will eventually, since the pH is reduced, it will lead to protein denaturation and destabilization of your proteins, and will also lead to the alteration of enzymatic activities. In addition to that, your ATP will be degraded into IMP and eventually into hypoxanthine. Now, these are the enzymes that's responsible for the degradation of your ATP. ATPase from ATP to ADP, myokinase from ADP to AMP, AMP deaminase from AMP to IMP, and monoesterase from IMP to inosine, and nucleoside phosphorylase from inosine to hypoxanthine. Ayan. So take note that not all the degradation products of ATP are negative or hindi maganda. ATP, once it's being degraded to IMP, it gives a characteristic fresh seafood flavor of the fish. However, as ATP degradation proceeds, it produces a product that is known as hypoxanthine, and this gives the fish a bitter flavor, bitter of flavor. Ayan. So this shows the degradation pathway of ATP. So again, from ATP to ADP, AMP, IMP, hypoxanthine, and ayan, inosine, hypoxanthine riboside, and hypoxanthine. So we have to note that AMP here is the desirable product of ATP degradation because it gives the fish its characteristic flavor. However, once this hypoxanthine accumulates, no, the odor of the fish, it gives the fish an bitter taste and an off odor na smell or odor, yun. So note that ATP and related compounds are broken down by endogenous enzymes as shown in this slide. And the flavor of fish are best kumbaga immediately after catch and up to the accumulation of IMP. However, IMP production or formation is very rapid. And mabilis lang siya. So, very rapid lang yung sweet and meaty flavor ng fish. And eventually, magre-result na into accumulation of hypoxanthine na it will give the fish a bitter off flavor. So, yon, very rapid itong IMP formation. So the degradation of ATP catabolytes proceeds in the same manner with most fish, but the speed of each individual reaction from one catabolite to another greatly varies from one species to another. So often, incidentally, with the perceived level of spoilage is determined by trained analysts. So kailangan din na trained yung analyst natin, no? That's why usually in researches, kapag merong mga sensory evaluation activities na involved sa project, they get a trained panelist, and also as much as possible throughout the study, the same set of panelists will examine the product. Ayan. We mentioned earlier that the living fish has a neutral, close to neutral na pH, but after death, and with the accumulation of lactic acid, the flesh becomes more and more acidic. So once it becomes slightly acidic, the texture is also becoming soft, and the flesh, however, at very low pH, the fish will become firm and eventually tough. Kasi 'di ba, if you would notice, if you put vinegar sa muscle tissue, it becomes white and nagtataffen yung mga meaty products natin. So it's the same with our fish. All right. So this slide naman shows the TMAO degradation process. So TMAO is degraded in two different pathways. So meron siyang bacterial meron siyang degradation caused by the bacteria, and another one is caused by the enzyme. Okay, so in the case of bacterial spoilage, no, caused by bacterial spoilage, the gram-negative bacteria growing in fish and fishery products, including Shewonella species, Photobacterium, Aeromonas, and Enterobacteriaceae, now are the ones that are able to use TMAO as an electron acceptor. And they reduces TMAO to TMA, to DMA, and to formaldehyde, no? So that is supported with the study of Summers in 2016. Those are the microorganisms that were isolated from fishery products, no, that were capable of degrading TMAO to DMA and monomethylamine. While in the case of enzymatic TMAO degradation, no, it is caused by the enzyme TMAO demethylase or TMAOase and TMAO-ASE para maging enzyme siya. So this is an enzyme responsible for breaking down TMAO into dimethylamine and formaldehyde or FA. And this enzyme is most commonly found in cod species or cod family, and this observed enzymatic spoilage takes place at 0 to -30 degrees storage temperatures. So even though nasa cold temperature siya or frozen condition, the fish still undergoes TMAO degradation. And note that TMAO degradation products results in the development of unpleasant fishy odor. Ayun. So how do you measure TMAO or degradation of TMAO? That is by the measurement of the total volatile base nitrogen. Okay, so and it is usually done by a Conway microdiffusion method. So we will discuss that in the next module. Okay. Di ko ma-move yung aking screen. Sorry. At hindi siya nag-move. Ayan. All right. So this is the summary of autolytic changes in chilled fish. No? So these are the substrates, and these are the enzymes that's responsible in the changes that fish undergo. No? So we have the glycolytic enzyme. So it produces the lactic acid, resulting in the gaping and reduction or reduction of the tissue pH. And then we have the autolytic enzymes responsible in the nucleotide breakdown, the ATP, ADP, AMP, IMP. And this results to the loss of fish flavor and the gradual production of bitter taste. We also have catepsins, no? And the substrates for this are your proteins and peptides, as well as for chymotrypsin, trypsin, and carboxypeptidases. So these are your proteases. No, they degrades your proteins into shorter amino acids. Ayan. So this results to the softening of tissue and also autolysis of the visceral cavity of fishes. Ayan. We also have calpains and collagenases. So ito naman yung mga substrates. And we have your TMAO-ase or TMAO demethylase. And the substrate for that is TMAO. And it results to the toughening of frozen ganoid fishes. Ayan. So very, very informative ang slide na ito. Okay. So next is we have the changes caused by microorganisms. So the bacteria penetrate the muscle of the dead fish, as what we have mentioned earlier. No? So they multiply quickly and produce metabolites that are responsible for spoilage. So yung mga metabolic products nila are the ones that causes or that manifest mga sensory changes that can be detected, no, as the fish undergoes spoilage. And also bacteria and its enzymes, no, kasi the bacteria are also capable of producing enzymes, change fish odor and flavor to sour, gassy, fruity, and finally ammoniacal and fecal na mga odor. Okay, so what are the effects of careless handling? It results to bruises in fish flesh. Also, it results in broken skin and bursting of the guts. And all of this, since there is an opening or the exposure of the tissue to microbial attack, this will now result to undesirable changes. For the first one, darkening caused by burst blood vessels, broken skin provides entry for bacteria, and burst guts becomes attractive to bacterial and enzymatic contamination. Ayan. So this one briefly shows the principle of bacterial spoilage. And we already have mentioned this, no? So I don't think we have to really elaborate more on that aspect. Okay. However, we have to note that fish spoilage is a result of microbial growth, no? And becomes evident as visible growth, no, ang molds, pigmented or non-pigmented, slimy bacterial colonies. So we know that there is invasion of microorganism kapag makita natin yung visible microorganism growth in our products. So in such a case, of course, there is a direct relationship between the total number of microorganisms and the degree of spoilage that your fish have undergone. Ayan. So for protein degradation, ito yung mga products. For carbohydrates, we have the alcohols, acids, and gases. For fats, we have the fatty acids and glycerol. And all of this, now if we're going to sum up the degradation products and we collect all of them, it will result to undesirable texture, flavor, odor, and appearance of your fish. Ayan. So this one naman shows the, briefly shows the principles behind bacterial spoilage. So we have the SSO and also we have your total count. So as you would notice here, your total count is always higher or greater than your SSO or your specific spoilage organisms. So your SSO is actually a subset of your total count. Kaya it's always lower. And only, di ba, your fish has indigenous microorganism or natural flora that is initially present kahit buhay siya. And once it dies, no, it proliferates and multiplies in the tissue. And there will be some SSOs or specific spoilage organisms that will grow in the product. And those will produce mga spoilage byproducts that can actually eventually be used as indicators. All right. So what is the meaning of SSO? What are SSOs? No? SSOs are the specific spoilage organisms, are the ones that produce metabolites that cause the undesirable odors and flavors associated with spoilage of fish and fishery products. So, yan yung brief description natin ng SSO. Ayan, so these are the SSOs, no? For chilled, for products that are stored at chilled temperature, we have your Pseudomonas, Achromobacter, and the Flavobacterium. For the shelf-stable products that are stored at atmospheric temperature, now we have your E. coli, Proteus, Serratia, Sarcina, and your Clostridium. And at high temperature na condition, we have your Micrococcus and your Bacillus. Okay. All right, so bacterial flora on newly caught fish depends on the environment in which it is caught, no, rather than on the fish species. So we also have a question in your quiz there, wherein we compared the natural flora or the initial microbial composition of fish caught from temperate water and from tropical waters. Ayan, so fish caught in very cold waters carry lower counts, whereas fish caught in warm waters have slightly higher counts because, well, there are more microorganisms that are more adapted at a higher temperature, and at higher temperature, diba, they are that's the optimum temperature for growth of most microorganisms. All right, so bacteria varies with raw material, no? Within the raw material, iba yung microbial count ng skin, gills, and gut, no? And then bacteria also varies with the environment, air, soil, and water, and also with the process involved, no? We have the equipment, the staff, the personnel handling the fish, and also the presence or absence of pests. So ayan. Take note, not all bacterial population initially found on the fish will cause spoilage. Kaya nga we have this total microbial population. Only those few populations called the SSOs produce metabolites which cause quality changes in fish. No, and these changes are important and can cause the product to be rejected. All right. Okay, so as prescribed by FAO, no, these are the set limits for fresh, chilled, and frozen fish products. Ayan. So the total viable count should not exceed, no, 5 times 10 raised to 5 per gram of sample. Okay? And the E. coli should not be more than 100 CFU per gram. And take note, Salmonella, Shigella, and Vibrio cholerae should be present. I should be absent. Should be totally absent in 25-gram sample. So that's how strict, no, ang implementation natin ng limits when it comes to your fresh, chilled, and frozen fish products. So if the total viable count, regardless kung madami ba siyang SSO or konti, should not be more than 5 times 10 raised to 5. Okay? So, ayun. So, these are the SSOs that should be monitored. No? The E. coli, Salmonella, Shigella, and Vibrio cholerae in your chilled, frozen, fresh, chilled, and frozen products. Okay. So, I think we're done with this one. The MAO degradation, bakit siya bumalik? Ayan. So, kasi we mentioned about bacterial degradation. So, TMAO degradation is also one of the pathways or one of the means TMAO is degraded is by bacterial action. So, ito yon. Okay. And take note, Shigella is one of the SSOs, no? Or is one of the gram-negative microorganism that's responsible for degradation of TMAO. Ayan. So this one shows how histamine is formed, no? We know that histamine is formed only in selected species of fish. Now, those species which have high histidine content. So, certain bacteria produce the enzyme histidine decarboxylase, and this enzyme reacts with the histidine. No, histidine is a naturally occurring amino acid that is present in large quantities in selected fish species. And example of those species are your tuna-like species, no? Yung mga scombroid species natin. Okay? And the result is the formation of scombrotoxin or the histamine. So, scombrotoxin is a foodborne toxin most associated with the consumption of certain species of fish like your mackerel and your tuna. And the histamine here, that is a biogenic amine, can be produced during storage of fish and certain other products, usually being acted on by the microorganism or spoilage microorganism. So yan yung concern natin when it comes to histamine. So histamine will also only be formed once there is histidine. So if the fish has low to no histidine content, no, very minimal histidine, hindi rin tayo magkakaroon ng problem sa scombrotoxin. Okay? So histamine in some seafoods, no, ito yung mga known histamine levels in some seafood products. So pag tuna, mackerel, talagang very significant yung amount ng histamine, no, to a level that can already be detected and can cause allergic reaction to some individuals. But squid, scallops, and prawn have minimal or low levels of histamine. Your sardines and also sardines and chanos milkfish can also have certain levels of histamine if those are not handled properly. Okay, so allergy, it causes allergy-like poisoning, histamine poisoning, and consuming scombroid and scombroid-like marine fishes with high levels of histamine can result to histamine poisoning, and it usually occurs at 200 ppm or more, and the severity depends on the individual and the presence of other amines and diamines. Yan. So these are the species, no, that can cause scombroid poisoning. So we have your tuna, bonito, and mackerel. Ayan, Clupidae, your herring and your sardines and your mahi-mahi. So take note that these species have high free histidine content and which may be converted into histamine during improper storage and during poor handling, especially if you subject your fish, your tuna fish species to temperature abuse. Ayan, and take note that histamine cannot be destroyed by cooking. No, it is heat stable. It's a heat stable toxin. And take note also that once it is present in the fish or in the fishery products, it can no longer be removed. So ang best means on how we can control histamine is by controlling its formation. Kasi once it formed, hindi na siya ma-remove. So dapat at the start pa lang ng processing, you already know how to handle your fish in such a way that you don't trigger histamine formation. Ayan. So we have these mga parameters, no? Fish products from fish species associated with high amounts of histidine, particularly fish species that were mentioned earlier, should have these results, no? 9 ang samples na kukunin per batch, and then those from that 9 samples, no? Dapat not more than 2 of that sample will have a reading of more than 100 milligram per kilogram na histamine content. And once merong umabot ng 200 milligrams, automatically i-detain na yung it will be subject for detention ang product. Okay? Fishery products which have undergone enzymatic maturation treatment in brine manufactured from fish species associated with high amounts of histidine can have as high as 400 milligram per kilogram or below na histamine content. Ito yung maximum level. But for fishery products that's usually sold fresh, no, ito yung magiging limit for histamine level sa EU market. All right? Now let's proceed with the chemical changes in fish, no? So we have lipid oxidation and hydrolysis. So, lipid oxidation results to the reaction that gives rise to a variety of chemical and physical changes in lipids. So we know that fish has high amounts of PUFA or polyunsaturated fatty acids. And those unsaturated fatty acids are very reactive and prone to auto-oxidation. Ayan. So, auto-oxidation is acted on by your lipoxygenases, and this results to the generation of odor and flavor in fish later on pag na-oxidize na yung PUFA sa fish, no? And hydrolysis also results in the formation of free fatty acids. And normally, this does not cause problems in fish, but causes an off flavor in oils or nagiging soapy yung flavor and odor ng fish natin. Okay? So, the two major deteriorative changes in fish which occurs in fish are the enzymatic hydrolysis of lipids to produce the free fatty acids and the glycerol, and as well as the oxidation of fish oils, yielding the rancid odors and taste, which are the major problem encountered in fish, no? Especially yung mga stored na oily fish natin. So yun yung mga ketones, aldehydes, and other lipid oxidation products are the ones that's responsible. Mga secondary lipid oxidation products are the ones that's responsible in the off odor and off flavor of fish. Okay? So take note, ito yung mga importante nyong malaman, and you have to remember this always, no? That fish has high amounts of polyunsaturated fatty acids. And these polyunsaturated fatty acids are very unstable and very reactive. And yon, very prone sila to oxidation. Ayan. So that's all for this discussion about pre and post-mortem changes. No, I will be posting this recorded lecture in our LMS under Module 3. So that's all for now. Thank you, and I'm sorry I can't turn on my video. Bye!